What is the genetic code? What does it code for? And did it arise all at once or did it evolve gradually? We will try to answer these and similar questions in today's episode of the series Secrets of Genes, in which I welcome you. My name is Eduard Kejnovsky and I work as a geneticist at the Biophysical Institute of the Academy of Sciences of the Czech Republic. We often hear in the media that the human genetic code has been deciphered, or that scientists are trying to decipher the genetic code of some organism. That's all wrong. This is an imprecise way of speaking by editors or journalists. They're not actually talking about the genetic code, but about the genetic information of the organism. Because the genetic code is something else entirely. It is a set of rules that determine how genetic information is translated from DNA into proteins, that is, how the sequence of nucleotides in DNA is converted into the sequence of amino acids in proteins. The genetic code was deciphered once and for all in the 1960s, in the 20th century. So please, let's not confuse the genetic code with genetic information or the genome of an organism. Let's take a closer look at how the language of 4 letters, the language of DNA, can be translated into a language that uses 20 completely different letters, the language of proteins. Each of the 20 amino acids that make up proteins had to be uniquely specified by several letters of hereditary information stored in DNA. How many letters? How many letters are needed to describe 20 amino acids when we have only 4 different letters available? If amino acids were determined by just a single letter in DNA, we could only use 4 amino acids, that is 4 to the first power. The aforementioned American physicist of Russian origin, George Gamow, stated long before the discovery of the genetic code that even a 2-letter code would not be sufficient, because it would only code for 4 to the power of 2, that is, 16 amino acids. He therefore proposed a 3-letter code. His prediction was correct. A triplet of DNA, or rather RNA, letters that codes for 1 amino acid is called a codon. This is something that was mentioned earlier. Given that each codon is made up of, there are 4 types of letters, and with triplets there are a total of 64 codons, or 4 to the power of 3. Since there are only 20 amino acids, many codons are left over. The code is constructed so that some amino acids are coded by more than 1 triplet of letters. Therefore, we say that the code is degenerate. The degeneracy of the code is a very useful thing. If a mistake occurs in the DNA, a mutation, and 1 letter changes to another, often the altered triplet of nucleotides still codes for the same amino acid, and therefore the protein does not change. The ability to choose different codons to specify the same amino acid actually allows additional information to be stored in the DNA, for example, about the 3-dimensional structure of DNA, such as the speed of protein synthesis, DNA bending, the binding of DNA to the cell's protein scaffold, or many other types of information. It seems that DNA contains multiple layers of information superimposed on each other, many of which we either do not yet know or are only just beginning to suspect. And only one of these layers determines the sequence of amino acids in proteins. That is why some scientists even consider the possibility that the code preceding today's 3-letter code might have actually been a 4-letter code. This would have increased the degree of degeneracy even further, making the code more resistant to potential errors. Such a code would in fact be more robust. When proteins are being synthesized according to the sequence of letters in the nucleic acid, it is necessary to specify exactly where the elongation of the protein should stop. This is the role of a triplet of letters that do not code for an amino acid. Instead, they specifically signal the termination of protein synthesis. They are called stop codons, and for extra security, there are 3 slightly different variants. The stop codon UAG is called amber, the codon UGA is opal, and the last one, codon UAA, is called ochre. how the codons got their names is accompanied by an interesting story. The codon amber was the first to get its name, given by its discoverers Richard Epstein and Charles Steinberg, who named it after their friend Harris Bernstein. His last name means amber in German, and the remaining two codons were humorously named opal and ochre to maintain the color-themed naming convention. Scientists are simply playful creatures. So, to sum up the main points briefly, stop codons halt protein synthesis in the ribosome. It's fascinating that the same code is used by viruses, bacteria, and humans alike. That's why, at first, people spoke of a universal code, indicating a unified organizational principle for all living things, in line with Darwinian ideas about the evolution of life. From simpler forms to more complex ones. It was therefore a big surprise to discover that there are exceptions. It turned out that in small cellular organelles called mitochondria, which serve as the cell's power plants, a genetic code is used that differs from the universal code. Even though the genetic code was deciphered more than 50 years ago, it still remains a mystery surrounded by many questions. Did the code arise all at once, or did it develop gradually? Were all of today's 20 amino acids, which now make up proteins, present at the beginning? Or did the first proteins make do with a smaller number of amino acids that were available at the time? On one end, there is the hypothesis of random freezing, the so-called frozen accident, which assumes that the code arose once, more or less by chance, and then did not change any further. Because even the slightest deviations from it would cause such profound changes in proteins that they would signify certain death for the existing organism. Given that alternative genetic codes also exist, there would have to have been multiple moments of freezing, which seems rather unlikely. And so, not only for these reasons, the idea of a random freezing is now more or less dead. And thanks to the amount of evidence, the prevailing view today is that the genetic code evolved gradually. The genetic code underwent a gradual evolution. And that's all for today. In the next episode, we'll talk about viruses that have survived from the very beginning of life on Earth, about living molecular fossils, and also about evolution in a test tube. I'm looking forward to seeing you. If you enjoy my storytelling, I'd appreciate a like. And if you'd like me to continue making episodes about genetics and evolution, please subscribe. That will help this video reach more people. I really appreciate your support. Thank you, and bye-bye.